Welcome to Translating Proteomics, where we explore the science of proteomics and its growing impact on basic biological research, drug development, and more. In this episode, we cover proteoforms and specifically discuss what proteoforms are, the challenges of measuring proteoforms, and the power that comes from measuring proteoforms. And now your hosts, Parag, Malik, and Andreas Humer of Nautilus Biotechnology.
Today, we're going to be talking about proteoforms. Well, there are 20,000 genes. Those genes are spliced in different ways to make nearly 70,000 different isoforms. They're then modified with different post-translational modifications, things like phosphorylation and methylation. And it's been estimated that there may be as many as 6 million or more different proteoforms. These unique versions of proteins each behave differently, they're regulated differently, and they drive how biology functions. So today's topic, we're going to dive into why are there so many proteoforms? How do we measure them? How are they impactful? How do we think about them as a next generation drug target or diagnostic? And everything proteoform. So Andreas, I'd love to start with you. Just when you think about proteoforms, when was the first time? Go back to when you first thought about proteoforms as different than just proteins.
I think I heard the term proteoform maybe 15 years ago. And you probably know Neil Keller sort of termed that, created that terminology. I thought of proteins way before that, much more in a dynamic way. My own graduate work was focused on, oxidation patterns on proteins and how that actually changed their binding to their target. And so I always looked at proteins not the way that we look at it from a FASTA type of, file perspective, a, reading frame or, a number of exons. I always looked at it more as a template for nature to play with. Mm-hmm. And so I have this, I don't mind calling them proteoforms, but I think proteins, I've always considered proteins as a very dynamic construct and not necessarily as a product output from a gene.
Yeah, that makes sense. And in my world, when I was learning, I very much grew up in a protein structure function world. And so I thought about the structure of a protein as being this incredibly dynamic thing. And if you added a large phosphorylated negative charge to the left-hand side of the protein or the right-hand side of the protein or both, you fundamentally changed the behavior of this structure. You changed how it moved throughout the cell, you changed how it interacted with other proteins. And so that was but that fundamental belief in it's not just the abundance of the protein, it's the modification state of the protein, it's where it is, it's who it's playing with. If we think that of the world of proteins as needing to understand all of those elements, why has it been so? Why are we just now really experiencing a rebirth of interest in proteoforms?
Well, before I answer this, I think the key word you just said was structure and function. I think what people really need to internalize is the fact that the composition ultimately creates a structure, which ultimately creates the function. Mm-hmm. And I think the fact that there's many different versions of a protein is just meaning there's many different versions of structures, which means there are many different ways of proteins to interact with other targets in the cell. And I think that's really how we should think about proteoforms. And I often hear this conversation about, how many proteoforms are there? In fact, there's papers out there that discuss this. Well, I think it's, in general, a useful exercise to sort of like where are the boundary conditions, right? Trying to understand, is it billions, is it millions? I think ultimately it's not really important. I think what's important is how fast these proteoforms can emerge. So the rate of proteoform.
Oh, that's interesting. I guess maybe but if you just think about it mathematically.
Yeah.
If each protein has potentially 50 different modifications, and we've seen things like the work from the Steen lab looking at tau and flexitau and showing large numbers of modifications that can exist on tau. If all of those combinations, that 2 to the 50, are possible, that's a dauntingly large space to have to consider when thinking about, if the modifications at positions 1, 6, 10, 15 are different than the modifications at 2, 8, 37, and they're both equally prevalent, that hypothesis space is gargantuan.
It is, but again, I may not agree with you that this should be the focal point of at least a lot of people are thinking about it. I think it's more about the fact that proteoforms open up more functionality, and we should be really thinking about, when a cell or an organism tries to react to an external change, environmental change, right? How fast can the organism respond? And that's the rate of change that really matters in that point, right?
Yeah.
And this is why, as you already mentioned, up and down regulation of proteins can't be all the response factors that a biological system has. It has to be something that's quicker.
That's a really interesting point that we dive into more in our conversation on dynamics. But just briefly, cells need to react to things on different timescales. So for instance, if I'm in a shock condition because I'm running out of air or I'm running out of sugar, or another cell has come in my proximity, I may need to react as a cell in seconds. On the other hand, if there are larger effects going on, there's a tissue effect, that maybe can wait for hours to days. And so depending upon what time scale I need to react to my environment, I might have different levers I can pull. And changing proteoform heterogeneity, changing which proteoforms are up or down or where, is a really fast, potentially, thing that I can change, because phosphorylating something can happen very quickly.
I also think about this from a bioenergetic perspective. If you think about the fact that it takes about a 4 ATP equivalent to add an amino acid to a polypeptide chain, then typical protein probably has 300, 400 amino acids that would set you back about 12 to 1500 ATP molecules, right?
Okay.
At any point in time, the cell can only make about 100 million ATP molecules per minute.
Oh, so you've got a boundary.
You just can't make more, right? And that doesn't. So
kind of
that
like
is
having a bank account, and so
exactly.
you only have so much money in your bank account that you can spend on ATP.
So exactly, you can't endlessly making and degrading proteins, and so you have to find different ways to modify the function of a protein. And so I think that's where the proteoform conversation comes in. And I think you could almost take that description and say, how many proteoforms could a cell make given the energetic budget that you have in the cell, right? Proteases sometimes need an ATP molecule, or reducing your equivalent would be the same thing, right? Also consumes ATP. So I think that's probably where I would start trying to triangulate this.
So
number.
there are these fundamental limits in nature that are governed by energetic processes, perhaps also moving proteins sometimes takes energy, folding proteins using chaperones, loading proteins. So there are these costs to everything you're doing, and some of them are lower than others. Building a whole new protein is an expensive process for a cell.
That's right. And I think this is why the presence of a proteoform is probably not only most energetic, the most conservative way to change the cell upon a stimulus, but it also probably is a very elegant way to do it, because you need to think about the other side of the equation. So now you changed a protein or made a different proteoform. Now you need to take care of it, right? Because now you want to change that proteoform's activity.
Yes.
You need to either degrade it, or you need to dephosphorylate it. You need to, again, you bring it back in the original format, right? Or you degrade it. So again, that costs energy, right?
So I want to, one thing I'd really love to get your perspective on. So we've been talking so far under the assumption that proteoforms are important.
Yeah.
What's our evidence? Why do you believe that proteoforms are important?
Well, I mean, obviously the reasons I described from bioenergetic perspective. But I think the other part is that it is such an elegant way to change the structure and then have a completely different interaction with other parts of the cell, right? Or as you said, sometimes molecules or proteins in particular get, relocated within the cell.
Sure, but that's still conceptual. What evidence do you have, or what evidence have you seen from talking to folks in the field that really has convinced you that proteoforms matter?
I think there's a number of examples that we have. One that's probably important for a number of reasons. But for example, there is the process-specific antigen. There was a test developed for early detection of prostate problems, and it was essentially no longer recommended. It turns out that there is a particular proteoform that is actually more indicative of prostate problems. And the test that had been developed, basically measured all of the proteoforms. So I think when we did some work back, a few years ago on looking at the different, proteoforms of PSA, we recognized that it was actually a very particular glycosylated form that was actually indicative or could have been the diagnostic for prostate issues. And so I think we have, many examples in that regard. Yeah. And I think for people that study proteins, that, that they appreciate that.
Yeah, that makes sense. I guess for me, it always felt, on one hand, incredibly obvious. Like, well, of course, why would proteins have this capability? Why would evolution bother with all of this machinery if it wasn't going to use it? But on the other hand, if you look at the number of first papers looking at transcript changes relative to the number of papers looking at protein changes, relative to the number of papers looking at proteoform changes, very, very different scales.
Why? Absolutely, but it's also to do with the tool set we have, right? I mean, if you want to observe a proteoform, it is incredibly difficult, particularly if the protein is very large, right? With modern mass spectrometry technologies, we can look at, let's say proteins 30 kilodaltons and smaller, right? And we can look at their, composition exquisitely through fragmentation spectra. But even that, when the mixture or the heterogeneity gets much more complex, it is very difficult to separate them. And, a good example is if you just have a small change where you, essentially add a Dalton difference because you're going from, through isoaspartate, a small change, but a significant change in folding, for example. Yes. It is very difficult to detect. So I think it points towards the fact that we just simply haven't been able, to detect those in an easy way, right?
So it's not there's not a belief that, proteoforms don't matter. It's, hey, I just don't have a great way to measure them. There are these limitations on the existing tools.
Well, majority of biology is created, through immunoassays, and they're really probing a particular epitope. And if that epitope does not, change, or you don't have further ways to probe the protein, you will never detect the proteoforms, right? I think there's also, though, a flip side to the conversation I think we need to bring up, right? Yes. Which is, all right, so let's say we make a tool that allows us to do proteoforms or measure proteoforms exquisitely accurately. What is all the information that we are going to get?
Yeah.
How are we going to deal with this?
So you're taking a thing that's complicated, you're making it exponentially more complicated.
Yes.
And how do you, as you do that, you need more data to be rigorous and consistent in what you're looking at. And so that's the struggle that you're bringing up, is the more you measure, the more you need to measure.
The more you need to measure, but also the more you need to be able to explain. Because I've learned, people like simple answers, and complex answers are just harder to deal with. And if you want to publish a paper, for example, right? I mean, you have to have a good explanation why you see all these different things. And so, I think proteoforms will be incredibly exciting, but they will also, give us a lot of challenges, because we will have to find out the way that we, even communicating. How do you communicate in a diagram or in text, a protein that has, Yeah. 50 different proteoforms and how they change.
Yeah, I mean, 50 different proteoforms is actually relatively compact. Again, you can accomplish 50 different proteoforms with 6 or 7 different modification sites. Once you get to 10, 20, 30, which, for instance, proteins like EGFR have 65 plus phosphorylation sites, you're getting into billions of possible proteoforms at that point.
And if you think, a paper on a particular biological mechanism puts you to sleep, wait until you have a paper that describes 50 different proteoforms.
No, I think that would be really exciting. I think getting to that level of detail and really understanding that causal relationship would be really, to see such an elegant mechanism would be pretty exciting.
I totally agree with you. Yeah. And I'm looking forward to the point where we have those tools, and I think we need them.
So let's talk a little bit about, we've talked about how proteoforms exist. We've talked about why we're challenged today. What do we need proteoforms for? How are they gonna show up in the world?
Well, I think you mentioned the Alzheimer's as an example. I think let's just build on that. So it turns out that you can basically measure the typical certain phosphorylation sites in a protein called tau protein that obviously changes during the disease progression of Alzheimer's. I think what we see is that these particular phosphorylation sites are regulated in a way that may reflect actually the progression of the disease.
Okay,
right.
So there's a fundamental mechanistic aspect.
There's a fundamental mechanistic aspect, right? And I think when you look, or at least if you overlap the phosphorylation sites with some of the kinases that might be responsible for those, you can clearly see those kinases are from different biological mechanisms. And so I think what you're seeing on the tau protein is essentially a reflection of these mechanisms that
Sure, but what do I do with that? How is that useful? I mean, it's interesting. It's part of our world.
They may describe the causal, the reason for Alzheimer's, right? That they may describe what actually causes, Alzheimer's. I think what we're seeing in this case is often just the symptoms of it, not the cause, right?
Mm-hmm. Yeah, I guess I would argue that there are really 3 big areas that the proteoforms could be influential. I think you described really a mechanistic role. I would argue that maybe there's a world where we have proteoform-specific drugs, where there's a particular proteoform that is bad, and instead of developing therapeutics that are going after total protein, they're going after the specific proteoform to deplete it or target it for degradation. So I think that's a really exciting avenue. I think also on the biomarker side. So you mentioned PSA for prostate cancer. We've also seen some evidence with serum troponin for heart attacks as well, that there may be proteoform-specific applications there. So therapeutic development as well as biomarkers for disease characterization. And then I think there may be a third possible application, which is in the future when we're trying to engineer cells. We may want to engineer particular cascades to occur. So there may exist a molecular switching future that involves proteoforms.
That's an intriguing idea to sort of think about. Maybe we can engineer proteoforms that are much more specific than general proteins.
Yeah.
Right. That's a very interesting idea. I haven't thought about it. Yeah. Might be really cool.
Yeah. So I mean, you think, for instance, about the calcium cycle regulation in our body and how calcium goes into and out of our bones, and maybe there are particular proteoforms that we can drive into our bones in a way that are more likely to get trapped there, or drive calcium cycle to build stronger bones through these sort of one-way mechanisms. Again, we first need to understand proteoform biology. So what do we need to do that? What do we need to do to understand proteoform biology?
Well, if you say there's 6 million different proteoforms in a human cell, well, you need at least 6 million different ways to measure it. Maybe not that much, but I think you definitely need a tool that has, the resolution capability. And as I mentioned, mass spectrometry can do some of this, but as you go and have larger and larger molecules, they become much more complex to unravel in a typical fragmentation spectrum. So I think we need a new set of tools.
When we think about technology, so you mentioned some of the limitations of mass spectrometry. Picture for a moment what is needed in a platform to measure proteoforms. What do we need to be able to do?
So I think the first thing that comes to mind, I think we need sensitivity. Obviously, those proteoforms are not, highly abundant in many
They're
cases.
substoichiometric,
Yes.
definitionally.
Exactly, by definition. We might need single molecule detection capabilities in some cases for proteins that are low abundant in the first place.
Yep.
We obviously need a very wide dynamic range.
Yeah.
To be able to measure them, in the context of other molecules.
Mm-hmm.
I imagine, for example, that many complexes can be assembled differently, by adding a particular proteoform, right? And then you may have to be able to distinguish those from.
So not just saying that, hey, these two proteins interact, but really being able to say, oh, when I look at the composition of this pair of proteins, it's this proteoform, it's proteoform number 6005 for protein A and 423 for protein B.
Yes, and I think this is where the finer biology is happening. Correct.
Mm-hmm.
Yes. So I think we need that, and of course, it is not going to be super useful if you can do that on a fancy instrument where you do one measurement a day.
Yeah.
I think we need something that can be large scale. It has to be easily accessible. People will have to. I mean, really, what I'd like to see.
Yeah.
In my lifetime.
Yep.
If that's possible,
Mm-hmm.
I'd like to see every single Western blot or immunoassay replaced with a proteoform measurement.
That sounds like a fantastic dream. I'm with you on that dream. That sounds like a big lift.
Yeah, it is probably. But I think
But on the other hand, I do feel like at this moment we're seeing a tremendous groundswell of interest and awareness of the importance of proteoforms. We're seeing numerous articles spread throughout the community in different forums about how proteoforms are critical to biology.
Yeah, I think in general, people are starting to understand that measuring biology simply by assuming that every protein is the same and has the same structure is probably an incomplete picture, right? And I think I can feel that, like you, I can feel that groundswell.
Yeah.
All right, so then let me ask you the question: when do you think we are going to be able to routinely measure proteoforms?
Tomorrow. No, seriously though, I think we're seeing an emergence of really powerful technologies that are able to look at proteoforms at a level of detail we've never seen before. And I think it's imminent. I think it really is imminent that within the next few years, this is a capability that's going to spread throughout the life sciences.
So you agree with me? We should replace all the Western blots with proteoform measurements.
No, I don't. So the reason I don't is that I'm a firm believer that you should use the simplest tool matched to the question you're trying to ask. And so there are some questions where Western blot is the right tool for the level of detail of question you're trying to ask.
Correct. Yeah. Absence or presence of a particular protein. Yes,
Right.
I agree.
So I don't want to replace all the Western blots, just most of the Western blots. Today, we've had a great time chatting about proteoforms, and in particular, highlighting that they are likely incredibly critical to how biology functions, but at the moment, they're also hard to measure. And so much complex biology might still be hidden from us. But there's an opportunity for emerging, incredibly sensitive, but high throughput platforms to really crack open this new frontier of biology. Thanks so much for joining us. Look forward to seeing you next time.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.